Thank you for everyone who made the journey, because this is the first event of this kind that we're doing in the region, and we really didn't expect so many of you to come and travel, but we're very happy you did. Um, so, I am co-founder CEO of Glycome, and we're a health tech biotech startup that's based in the region, but also US, Europe, and the UK. And we're pioneering a technology that very few probably know about, that I'll tell you in a minute. But finally, our science was recognized by leading universities. We've been publishing with Harvard, Yale, Hopkins for the last 15 years. Together with them, we publish papers in top medical journals like Circulation, Nature, Diabetes Research, and we're recognized by top media like Forbes, Wall Street Journal, BBC. And we have one single mission, which is to make preventive healthcare possible for you. And the problem with healthcare today is that it's not healthcare, it's disease care. We only tackle problems when it's already too late, when you're at a state of diagnosis. But chronic conditions can develop over decades. We just didn't have the tools to detect them on time. And all of this starts with aging. And the tools we were missing are called glycans. And glycans are one of the 4 building blocks of life. We have DNA, protein, lipids, and carbs. Glycans are the complex carbs. They're coated all around our cells, all of our molecules and proteins, and they give our biology the power of communication. So we have multicellular life because of glycans. If they were not part of our biology, we would still be single-cell bacteria. And if you spend any time looking through a microscope at bacteria, it's not a very fun life. So I'm very glad we have glycans. A lot of times, especially medical doctors, confuse them with something called glycation, which is how your body responds to the sugars you eat. But actually today, I'm talking about glycosylation, which is the development of these complex sugars that we manufacture within our cell. They become a core component of proteins and lipids, and they give them functionality. They participate in every single biological process we have. For example, your blood groups are defined by glycans. Conception happens through glycans. Your immune system communicates and functions through glycans, and every other process as well. And it was over 3 decades ago that my father started studying glycans. In fact, this was back in 1991 when I was born, and this is how he met my mother. They were working together in a glycan research lab, a very early one at the time, and they published their first paper, which was on glycans and aging in the brain. So I guess eventually I was also going to end up in glycans, although I tried my best to do something very different. And within these 3 decades, they've managed to publish over 300 papers showing that glycans are the first thing to change up to a decade before you're diagnosed with the disease. And as far as we know, they're the only molecules of this kind that can be so early in detection. And all of this is published in top journals and was supported purely by grants. So it was 40 million in grants over 16 years of science. And not just that they change before disease, they also change with the aging process. And they actually pin an underlying aging process called inflammaging that I'll tell you more about as we go down. And glycan age, or the glycan clock, was one of the first aging clocks developed over 10 years ago. And it was actually published on the same dated year as the first epigenetic clock, which many of you might have heard of, because epigenetics are far more popular than glycomics. And just last weekend, we were celebrating the 10th anniversary of aging clocks in Rovinj, Croatia, together with Steve Horvath, Gordon, and a number of other scientists in the field, and Gordon's my dad. And that, about 3.5 years ago, turned into a first commercial product. So it's been around in science for over 10 years. We've been publishing a lot, but it was not commercially available. We made it commercially available. It's a blood test that can tell you your biological age. And we pair it with specialist physician advice to give you an action plan on how to improve it. And actually, we do have a colleague here today doing testing. So if you are curious, you even have a chance to do this today. And recently, there was a review paper looking at all of the aging biomarkers used in practice. And our clock was evaluated as the most predictive of disease in the future and most responsive to interventions. We published over 25 papers just showing responsiveness to different lifestyle and pharmacological interventions. And this is how it looks in practice. So I'll tell you about one use case, actually one of our first users, which was Tim Spector. And if you haven't heard of Tim Spector, he's in 1% of the most cited scientists in the world. He has a H-index of 245, which is very, very high. And back when this was a research tool, so this was 2016-15, he wanted to do a test. So we went into the lab, donated blood. 6 months later, a PhD student generated his result. And he got a biological age. He was 58 at the time. His biological age was 42. And then he tracked it every year. And you can see throughout the year how he ages about a year per year biologically as he's chronologically aging. And then in 2020, he did an experiment and he tried metformin. He tried metformin because he already had a bit of a dysregulated glucose response and he had a family history of diabetes. Although he wasn't pre-diabetic or diabetic yet, metformin really had an impact on him and it lowered 10 years of his glycan age. And you'll see now he's 62 and aging a year per year, but 20 years younger to what he should be. And why do we need this? Why is this important? Well, I wonder how many of you consider yourself a standard human that's due to follow standard health advice. And unfortunately, that's one of the biggest misconceptions. There is no one size fits all. There's no one diet, no one exercise program, no one drug, no one supplement that works for everyone. And reason is many of them, actually. So from genetics to epigenetics, metabolism, gender plays a big role in differences. And glycans are integrators. They combine all of these different influences and put them into one molecule that has function. So within a couple of months, you can evaluate if something has actually worked for you and will give you a good long-term health outcome. And you'll see a study here that was done in Edinburgh University that looked at if people have an accelerated biological age, how predictive is this of hospitalization in the future? And you'll see in the last bar here, glycans were the most predictive of future hospitalization within a 10-year time frame. So you're changing something which is meaningful for your health outcome. And the process we're looking at is inflammation. And inflammation is this vicious loop of inflammation that starts when we're young, our immune system loses function, and then turns into many different diseases. That's from cardiovascular disease, diabetes, cancer, an immune system which is not killing something it used to and used to be good at. The good news is we can also do something about it. So we've shown predictive power on all of these conditions, and unfortunately, it's majority of conditions. It's 3 out of 5 of us that will eventually suffer from them. And we've shown that many of these cases you can reverse with interventions, both lifestyle and drug. And I'll highlight 2. One is diabetes. So we can see that glycans change up to 7 years before you have insulin resistance. And this is still a few years away from full-on diabetes. And we've shown in papers with thousands of people that you can reverse this change in glycans with caloric restriction and weight loss. So we can tell you today that in 7 years' time, you're going to have a disease, and you can do something today and measure how well you've actually prevented that. The same is true with cardiovascular diseases. We see that glycans can change up to 10 years before you have a heart attack or stroke. They also change 5 years plus before hypertension. And we see these same glycan structures that predict the condition change with physical activity. So we can tell you that you're heading towards this problem in the next decade, and you can measure how well you're doing now. So you get the feedback when you need it, not already when you have a problem, because then there's very little we can do. And where it gets really exciting is here. So a few months ago, we published a review paper that looked at all of the different conditions that are associated with what we measure in the glycan aging clock. And we identified 72 different conditions, going from cardiovascular disease, autoimmunity, cancer, neurological conditions, and a category that we say non-disease, which is smoking damage, perimenopause, non-alcoholic fatty liver, and many other. And by now you're wondering, well, how accurate is it? Does it work? And there's one criteria to evaluate biomarkers accuracy, which is sensitivity versus specificity, or the AUC. And the closer your biomarker is to 1, the more accurate it is. And when we look at glycans, in majority of the cases, they add a lot of information to what you're looking at. So here you'll see PSA, which is a routine blood test, a protein level, to indicate prostate cancer. And the problem with this test is it's very inaccurate. So if you look at the AUC, it's 0.60. So half of the time, the test tells you have cancer when you don't have it, or that you don't have cancer when you do. If you look at the exact same molecule, so again, PSA, but you look at the glycans on it, so you understand the language, how is this molecule actually functioning and communicating, all of a sudden you get the AUC of 0.94, or one of the top performing biomarkers. And this is true for many different cases. And the reason it is because if we just look at proteins, we don't see the full picture. We use this analogy quite often, which is this naked bird. If what we're doing in clinical diagnostics now is mainly looking at naked proteins and naked lipids, and mainly we're looking at the level of them in blood. Well, if you add glycans and you understand flight, you understand functionality, all of a sudden you see a completely different, complete picture. And these are a few of our biomarkers. You'll see hypertension, ovarian cancer, Parkinson's, and breast cancer. The gold standard currently used in practice is the green line, and glycans are the orange. And you'll see that in every single case, we outperform the gold standard. And by this point, people ask us, well, why is it not used in clinical practice already? Why don't we have these solutions today? Why can't we utilize them? And it's a very simple answer. Glycans are the most complex thing to analyze in our biology. If we look at building blocks, in DNA we have 4 building blocks, proteins have 20 building blocks, glycoproteins have thousands of building blocks. So they're incredibly complex. It takes us 7 days of different chemical steps to give you one set of to run one set of samples and give you a result, which is way too slow for healthcare or clinical practice. But we're confident that in the next 2 years, we can bring this down to 1 day, which brings us closer and closer to clinical practice. And we are the best people in the world to do it. So just last year, we published a review paper, or review paper was published, that looked at all of the public human glycomes done in different clinical trials. And out of 191,000 published human glycomes, our lab generated almost 160,000 of these. So we have the largest data set, we're the fastest moving, and we have the highest accuracy. And all of these samples came from Harvard, Yale, Cambridge. These are precious samples that they've been keeping for decades, and they trust us to analyze them for them. And we're the only place they can analyze them, because they do. Some of them have glycan labs, but they're not high-throughput glycan labs. So if they want to look at thousands of people, they have one place to go so far. And our plan is to expand this to the Middle East, to the US, and other regions. And I hope that this gives you a bit of a picture of what glycans can do. And there was a criteria how we'll do medicine in the future, which is it should be preventative, it should be predictive, participatory, and personalized. And glycans give you all of these 4 criteria. So I really hope that we can transform how we do healthcare today. Thank you for your time. Do we have?
I have done about, I've stopped now, but there was a time when I was very interested, and I now know my biological age. I've done about 6 different aging tests, and they all showed very different numbers. This can be extremely confusing and could create anxiety in people who don't know themselves as well as I know myself, for example, because that's what I do, biohack. So I know it depends on the markers, but I don't believe that there is much truth in any one particular one more than on the other one, because they all claim what you claim for yours. So can you just shed some light on that, please? Thank you.
Problem with having so many tests on the market is that as a consumer, you generally don't read research papers. So a lot of times when they make a claim, validating these claims means going back and seeing if they've actually published something on a topic, because most of the time they're extrapolating something from somebody else's science, and sometimes even creating something completely new that applies the same claims as what was published. So all of our claims come from our science. We're not extrapolating anybody else's science. And there is differences. So if you're looking at different types of, like if you're looking at epigenetics and glycomics, completely different things. And in this study, what was shown, that both of these clocks can go in different directions. Now, there's no one gold standard to measure aging. We haven't yet found it. There's no one mechanism in aging. And because it's such a complex process, it will always be multifaceted. And some people might have a problem with inflammaging, which is our mechanism. Other people might have a problem with epigenetics. And different interventions would apply to different people. So at one point, you'll be looking at all the omics you can measure. You'll be seeing where is your particular problem area. Is it inflammation? Is it epigenetics? Is it something else? Is it lipids? And then you'll be applying interventions that work for those. So I think that's where the confusion comes from. But what I recommend is start from the worst one, see how to fix the worst one, and then work on that one. If you have a good one, that's usually a nice excuse for like a bragging rights, but actually the point is improving every biomarker we identified has room to improve.
Maybe I've missed this part, but I was wondering if you can tell us a little bit more about the demographics of the populations that the studies were done on, and what are the numbers and equals what exactly, and whether you had different grouping of the population, so you validated in people with healthy people versus people that perhaps had cancer or cardiovascular disease, just to understand that a bit more. Thank you.
No, absolutely. So the total data set we have is 200,000 samples. It's mainly biobanks and different trials. So in biobanks, you're looking at healthy people, and then as they age, they would have a follow-up. And they come from 31 different populations globally. We have a lot of diversity as well. We have 20,000 Chinese samples, for example. We have the British minority biobank within there. We have, of course, a lot of European biobanks, like Twins UK, Estonian Biobank, or CADIS. So it's very, very broad, both healthy and sick people, both cross-sectional studies and longitudinal studies. But this is as big as the data set in glycomics gets. There's no bigger data set than this 200,000. And there's a lot of room for improvement.
Information. Just want to know, are there any other medications apart from metformin? Because some people don't react well on metformin, they don't metabolize it.
So actually, metformin was an individual case. And we did a recent placebo-controlled trial on slightly overweight, healthy people. And metformin had no effect in this trial. So what we're looking for now is a pre-diabetic or risk of diabetes population to see if we see the effect, because not even all diabetics respond to metformin, and it's certainly not a drug for everyone. Estrogen is a very interesting one. So we have 3 different trials on estrogen, and all of them came back with positive results, and it's actually both in men and women. So we see that post-menopause, estrogen reduces biological aging in women, but we also see that testosterone replacement in men has the same effect if you're not blocking the conversion to estrogen. So if you give them aromatase inhibitors and it doesn't convert to estrogen, you see no positive effect. So it's actually estrogen in men and women. And there was a nice trial, the ATP, or the Aging Intervention Testing Program. They didn't test the regular beta-17 estradiol that's used in healthcare. They tested the alpha-17 estradiol that doesn't feminize, tested in mice, and it expanded their lifespan as much as rapamycin. So it was about 12 to 19%. So I would say the validated drug we have is estrogen. Of course, again, doses vary, doesn't work for everyone. There's lots of complexity in every drug. One more? Okay, let's do it.